All right, welcome back. This is chapter 2, uh, part 2, and what we're going to do is finish up on the media, and then depending on how much I blabbed, I might just continue on and finish out the microscope section that they have, and then I'll move right into chapter 15, which is a simpler chapter, and that should be done today. So, without too much, let's get going on this section here. So, selective media, I've already kind of mentioned that last time. And this is where we understand more of the basics of how the microorganism deals with the environment and osmotic pressure and those sorts of things. And we take advantage of that understanding, and then we can make media that's selective. So it only what we try to do is get rid of certain types, maybe just the gram positives and we get rid of gram negatives, or we want just the gram negatives and we get rid of gram positives and that sort of thing. And you can do that kind of strategy in a clinical lab. You get all sorts of things. You get these polyinfection type of agents going on. We have to kind of draw a line and figure out, at least from what we're getting and understanding what's causing the problem. And we can't do it all at once. We kind of have to do it in a selective fashion, so hence the media. So it contains one or more agents that inhibits the growth of certain microbe or microbes. And so it's important as a sort of a screening or primary isolation strategy. And what you can do is you speed up the isolation by suppressing the unwanted background organisms, favoring the growth of the desired ones. And there's several strategies about this, but I just wanted to kind of drive home. We can use the selective nature of the media, and you can see the general purpose versus the selective that's in this diagram, and you can see that there's fewer organisms on the selective because we've gotten rid of the others and on this plate, but you can see it. And then using a differential media, which we've gone over, you can see that with those that we've selected out and the subset, even those now show some marked differences. And this is a good strategy to use when you're trying to sort through the clinical mishmash that we might get from urine or a lavage of, let's say, an infected area that's abscessed or something like that. So a medium can be both selective and differential. And again, the McConkey auger suppresses the growth of some organisms while producing a visual distinction between those. So it's that selective nature using bile salts and crystal violet dye, which is we'll talk about in the Graham stain. It's essential. Graham, he was pretty smart. He didn't know what was going on, but he could reproduce his work. He must have been a meticulous note taker. But anyhow, we'll get to that. So McConkey auger is a good one, so you can see colonies that change colors and then maybe some that don't. But we use dyes as differential agents, pH indicators that change as a response to production of acid or base, all sorts of things that we can do if it produces hemolysin or not. We use blood auger and those sorts of things. And so there's the McConkey plates, and we've already gone over this, and the mannitol salts auger, which is a really nice one to differentiate staph and other species of staph like epidermidis and Staph aureus. Look at the difference from the differential point of view of the yellow versus the pinkish color, and it's strictly a pH thing where E. coli won't grow on a mannitol. So reducing medium that contains substances that absorb oxygen or slows the penetration of oxygen. And it's growth for anaerobic, those bacteria that grow without the presence of air. Now, this is a very common media. It's not really discussed. It should be because we use it all the time. Is transport media, and all that is a swab. Usually, the end of the swab is somehow affixed to the top of a tube. So when you pull it out, the swab end of it comes out of a media, some liquid media to keep it from drying out, and then you go and take your sample and put it back in so that the environment around that the swab will be protected, or at least won't allow it to dry, and you'll get a good sampling of what's there. And I like the swabs, and you can see here's a swab. And Stewart's medium containing a reducing agents to prevent oxidation. So if I know I'm going to uh, take a sample that may be a anaerobic, now going in and getting these samples, I remember wearing um, overalls, had these plastic or rubberized bib overalls, and then opening up the intestines within a dead cow to try to figure out what happened, or a dead animal, some kind of large animal. You go up to your waist in these guts and things. It's really nasty. I can give you more of a view of it, but I think I'll pass on that. But you can see that the swab is sort of in the media, and in this case, the Stewart's medium prevents oxidation, because believe it or not, some microorganisms are so susceptible to oxygen that you can kill them almost just immediate upon exposure. And those are really hard samples to acquire. Usually it's a lavage that you keep liquid closed off anaerobically, but some of them are not quite as fastidious as that, like the gonococcus, and we can put that in a swab, and the charcoal neutralizes various things that can cause that organism not to grow, and there's all sorts of tricks that we can play. So carbohydrate fermentation media is a very standard thing that we use. The sugars can ferment. Now, all that means, and we're going to talk about this, enzymatic pathways. Eventually we're going to go in and talk about bacteria and how they break down certain things, and in the process of breaking it down, then it provides it in a format or a smaller or a more useful product that they can use to incorporate in building their infrastructure, or to do whatever it is that bacteria want to do. And the tools that we use for this is, of course, the various different types of sugars. You can have lactose, you can have trehalose sugars, which is a really strange sugar. There's hundreds of different types of sugars that one can use. Alpha, or dextrorotary, lever rotary, ones which way the structure goes. And believe it or not, some bacteria can't use dextrorotary glucose. It has to be lever rotary or something like that. So there's a variety of things, and how we detect they use that is that they can produce gas. That's socially unacceptable, but the bacteria don't care. But anyhow, they do that, and you can see that in these little Durham tubes. These are smaller tubes within a tube, and it captures some of the gases that are produced, and because it goes to the surface, but it's captured within that Durham tube. All it is just another glass tube, and you can clearly see that there is a gas forming inside, but the bubbles, and there's a big bubble at this end, and it lets us know that it's producing acid. So you can see the pH change. We went from red to yellow. In addition to the acid production, we see the gas or no gas, and that lets us know what type of fermentation that we're dealing with. And there's certain microorganisms that do one or the other, and it's just one more piece of the puzzle that we use to identify it. Now, I want you to think about this, because this gives us a huge amount of flexibility, and that's where we get tested on our knowledge of microorganisms. E. coli that break down lactose where Salmonella doesn't, and things like that are really, really important, how we use a diagnostic test. Now, granted, I want to just keep reminding you that there are genetic tests that we do now. It's the DNA fingerprinting and that sort of thing, that makes the process a lot easier, and you don't have to do the manual types of things. But there are some labs that still do both, and genetics. A lot of times these traits it's important to understand how they may have the genetics but not express it, and that sort of thing. It depends on what you're doing. Over here are various different types of detectors. They're sort of like litmus paper, or there are chemicals that respond to changes in pH. Now we know the changes of pH. You've had that probably discussed several times. pH represents the percent hydrogen. So I always like to talk about pH in terms for my students that pH is the amount or concentration of the hydrogen. And so if you have 100% hydrogen down here, the pH is 0. If you have 0% hydrogen that's present, which is a breakdown, of course, product of H2O and hydrogen, and then the other half of that, which these are inversely proportional. So down here at 0, OH, hydroxyl group, would be 100%. Is that right? No. pH for at the 0 end, or the acid end, is 100% hydrogen, 0 hydroxyl. And so it's the breakdown of water. So at 7, you have an equal amount of the hydrogen and hydroxyl group. And you already know that. I just want to kind of put it in the terms the way I like to look at it. And then here is 100% hydroxyl group and 0 of the hydrogen. So what we have are chemicals that when you put these are usually papers, or it's a chemical that it's in a liquid, and the chemical is always vibrating. You know, if it's in a liquid, it's vibrating, and that concept is from physics class. But the idea is these molecules are floating around and they're shaking, and when light hits it, it rotates the light in such a way that we perceive it as color. And so the frequency is really what it's changing. So by the time it hits our eye, whatever this chemical has done, it's vibrating in a pattern that we see and recognize, or at least the pigment in our eyes recognize as a color. So we have the cones in our eyes, and various pigments will start vibrating. It fires a neuron, if you remember that from anatomy, and then that's associated with a particular color band, and then our brains interpret it as a color. And so as the pH changes the chemical structure, eventually, of course, the pH will, because of charge, and the compound will start to shift a little bit in its shape. And when it does so, it may change its rotation or vibration patterns for a different color. So it moves into a slightly different spectra. So you can see, like thymo blue, we go from a red to a purple, and there's a region that's yellow, and so you can see a transition, which is really nice to be able to use these types of things. Like the phenolphthalein, it's normally doesn't have a color until it sees more of the higher end change of pH, and which is nice. You know, they did that in swimming pools. You can put phenolphthalein in the water, and you can detect to see if people are urinating in the pool. And I'll just let you know that the pools immediately would change colors, indicating that, well, you know, I think no matter where we go, we're going to see urine in the pool, so we're not testing for it anymore. Just a tip from your Uncle Ed there about that. So anyhow, we have various dyes, substances of pH indicators that we can use, and based on the pH, its structure will change, its vibrational pattern will change. And don't worry if that kind of escapes your understanding, but that's okay. I tend to like the visual spectra and the understanding of rotation of light and vibration patterns. It's really interesting to me, and so that I like to know about those sorts of things. So there you go. And so you can see when microorganisms break down certain, let's say, glucose, and it goes through metabolic pathways, which has a series of enzymes that all enzymes do is reduce the amount of energy it takes to break down. It's not consumed in the reaction. We'll talk more about enzymes, but you've already had that. But enzymes are not utilized in the process, but what it does is it greatly reduces the amount of energy it takes to break things down, and which is important. Energy, of course, is in the form of a chemical bond, which we go from ATP to ADP, and we release energy, or we can take energy and capture it and put it back in. That's what's so cool about ATP, adenosine triphosphate. Anyhow, uh, so the idea is you have enzymes that go from like pyruvate to acetyl-CoA or whatever. The idea is at some point in process you'll form like a lactic acid, which is an acid which will change the pH. And when it does is the lactic acid will interact with the indicator and change that particular pH structure of the indicator, and preponderance of that is you'll get a color change, so you can see the idea. So, and you can see the changes in color and things based on the vibration that is altered by the pH. Okay, so miscellaneous media. Assay media are used by technologists to test the effectiveness of antimicrobial drugs. And we do this, there's two I'm going to mention. Don't worry if it escapes you right now. But we use the antibiotics, of course, to treat, to get rid of. If somebody has an infection, we use an antibiotic. And most of the time, the bacteria have acquired the characteristic of being resistant. That's the problem we're having right now. But there are still some that we can utilize an antibiotic and treat, and they don't have the genes or the genetic potential for that yet. But the other is that we can use it for identifying, because some microorganisms have a constant or consistent characteristic of antibiotic resistance, and it's on the chromosome of the bacteria, its main big circular piece of DNA. Now, bacteria have smaller circles, like those little rubber bands that you put on braces for teeth. I never had braces, but I've seen people mess with these little dinky rubber bands. And think of the main chromosome as a huge rubber band, and so the smaller rubber bands represent little characteristics genetically providing, let's say, ampicillin resistance or some other antibiotic, and those are little circular pieces, or the small rubber bands, or plasmids. That's what we refer to them as a microbiologist, and we'll get into that. But plasmids are very common, but they tend to be unstable. In other words, they can lose the characteristic or that sort of thing. Bacteria, if you don't need it, they lose it. You don't need to have it. It's sort of like learning a foreign language. If you don't use it, you lose it. I found that out a hard way. But anyhow, so we can use antibiotics. Sometimes we can use disinfectants and other things to test to see how an organism responds to it. Also used by industrial and environmental microbiologists, we need to count the number of organisms in milk, food, and there's tricks for that. So you grid it out, so you show the bacteria, let's say, under a microscope slide, and instead of counting every one of the things, you have a grid, and you count up 1/4 of the grid, and then whatever that number is, you times it by 3, and now you've got all of them counted for. And it's just a way to work smarter, not harder, type of thing. So based on the concept that if an individual bacteria cell is separated from other cells on a nutrient surface, it will produce a discrete mound of cells. And I've already talked about this, and this is the bacteria colonies that we see, and it's just a piling up. Some of them are dead, some of them are alive, and a colony is defined as a macroscopic, large, viewable cluster of cells appearing on solid media arising from multiplication of a single cell. And so you can imagine the sheer numbers of these things going on. So the isolation requires the following: a medium with a firm surface, an agar plate that's been poured and cooled in a petri dish, and then you take an inoculating loop and you streak it on the media, and I've mentioned this before, and then you get the colonies that show up. So you have an isolation. You can have some sort of differential plate, and you can see the differences between the two, or you can put selective media and just grow one or the other, all sorts of things, and you can see the colonies showing up. And it's a very common laboratory type of deal. So steps in streaking a plate, and we're going to go over this. It's one of the labs that we'll be doing. and the loop, and so you see the loop. Now, just I pointed out now, a loop has, it's planar. In other words, if you go up and down, it becomes a cheese grater, but if you make it flat or longitudinal, then you can use it to make a nice smooth sort of surface at which to transport bacteria onto the surface of the agar. This is a good single colony isolate showing all sorts of messy contaminants or other organisms that are in there. You have yellow and red ones and white, and it tells you. And then you can dilute and on a pour plate, or you just make dilutions and you can enumerate them that way, and you, whatever your dilution factor is, and you can use that. So this would be countable, so you'd have like 20 some microorganisms here, and then depending on the dilution, let's say this is 1 to 100, 1 to 100, so that would be 10 to the fourth, and I said we had 20 here, so it would be 2.0 times 10 to the fifth. These would be each of these would be 100, so that's two zeros adding up to 4, and then there's 20, so there's a 0, and we always represent numbers as a single digit point as one of the labs that we went over. So it would be 2.0 times 10 to the fifth, and that would be what the original concentration was after we do the dilution. So a spread plate, we can pour the bacteria on a plate, and then we use what's called a hockey stick. It looks just like a hockey stick, and usually we have a platter underneath it and spin this, and then you can evenly distribute those, and so I'll show you some videos of that. So microbes can be identified through microscopic appearance, and we usually do that to confirm whatever we see on a plate. Characterization of cellular metabolism, so we let the bacteria tell us more about them. You know, they have to tell us because they're going to grow, and then we'll see what they can metabolize. Determination of nutrient requirements, products given off during growth, presence of enzymes, and mechanisms for deriving energy helps us identify. And then with the newer genetic and immunological characteristics, we can identify it and confirm even faster and better. And so that's kind of where we're at. So converting amongst the different units of metric system is important. Listing and describing the three elements of good microscopy, which we're going to talk about in the next section. Differentiate between the principle of light microscopy and electron microscopy. Electron, we get out of the limits of light. About the highest we can do magnification is about 2,500 times the magnification. So if you go to a store and you get let's say just a little magnifying glass, that will be about 30x. If it's a nice quality one, maybe 300x. And so you can see that, and we're talking about with a light microscope about 2,500x. And so that's about it. Electrons, though, electrons allow us to do a little bit different. Light limits the amount of light in a microscope, limits its ability for the resolution. Well, electron microscope allows us to resolve much better and get huge better. We can have 10, 100,000 times magnification using electrons, and I'll show about that. But we also use stains to denote. Bacteria are clear for the most part. They're kind of boring. If you look at them, you might not even see them, but we use a stain as a way, what they call a vital stain. The stain actually sticks to the bacteria, and it gives us sort of like painting them a color so we can see them better under a microscope, and that's what we do. So let's go on to the microscope. I'm not going to stop. I'm going to blast right on through here. The size of the macroscopic versus microscopic. Here is some which we learned in the lab, one of the labs, the first, I think lab 1, giga, mega, kilo, and then you have the base unit meter. In this case, we're talking about the length of something. And we don't use these terms up here that much on micro. Obviously, microbiologists are more involved with smaller things. So with the meter, we go on the other side of the meter and we start looking at centi, which is 0.01 of a meter. And then there's millimeter, which is 0.001 of a meter, and we use the terminology milli or million. Micro is 10 to the minus 6, so if you can see that it displaces the 1 by 1, 2, 3, 4, 5, 6. So that's 10 to the minus 6, and that's micro. And that's a good term for microbiologists. And then nano, this is the age that you're living at, and nano in Latin is 9 and 10 to the minus 9th. And again, if you count the displacement of 1, it's 1, 2, 3, 4, 5, 6, 7, 8, 9, and then the 9th, so it would be 1.0. So it's just counting the zeros across, and that gives you the exponent of a factor of 10, and then all you're really doing is the number of zeros. When you indicate with a negative number, it now goes on the other side of the decimal. If it was a positive number, the zeros would be going on the other side. And I'm sure that most of you are familiar with that terminology. And so anyhow, follow along with that. Microbial size, protozoa and algae usually about 3 to 4 millimeters. And if you look at have a micrometer or a ruler with millimeters on it, you can see that. Yeasts are generally 3 to 4 micrometers, so they're much smaller. Micro is smaller than milli. The smallest bacteria measure around 200 nanometers, and that really pushes the limit of our microscopy. In fact, we have to put oil on the microscope to get, typically our scopes top out at around 1000x. And the oil, by bending light, we get more light into the microscope. I already mentioned that light determines the limits of a scope. And we can add just a little bit more light using refractive index gain. And we get about 250x more magnification due to the additional light bent into the body tube of the microscope. And you'll get about 1250x or 1300x depending on your scope. And that's pretty good, but that allows us to see this somewhat in good resolution of 200 nanometers. So I'm thinking staph or strep, we typically have to do that. Most viruses are beyond the limits of a scope, a microscope. Using light, we have to go into the realms of electrons, and I'll show you some of those as we go on. So the size of things in a pictorial point of view, you can see a red blood cell is around 30 micrometers. We can go down to E. coli is around 1 micron, and I've already mentioned a micron, or another way to look at that is 200 nanometers, and so I just changed the unit a little bit. So we're looking at, this is about 100 nanometers, and E. coli is small. Staph is sort of right in there, so we have to use oil. It's right at the limit, the edge of light microscopy. And then we go into the range of electron microscopy, and now we can open up and see mycoplasma, viruses of various types, even visualize proteins. And then we can utilize a special microscope, an atomic force microscopy. We can actually look at atoms and amino acids today. In fact, I looked at a unit that I could put in the stem cell. If I wrote a grant, I could pay about $40,000 and we can get an atomic force microscope, which allows us down here, and I'll show you about that. So it's a good range of microscopes these days. So you see the light microscope. This is an electron microscope that looks like a Joel. It's a brand of microscope, electron microscope. It's a nice looking one there. So here's the scope. Now, most scopes are the same, and they may be a little bit better at that or not. The scopes that we have in the lab and the scope that I'll be showing you for this class is one that it has some nice characteristics. So you can be viewing and a slide, and then you go up in magnification, and when you do that, it'll be somewhat in focus. And they call that parfocal, and it's a really nice characteristic. So there's some added features of alignment and careful recognition of the power of the lens, that you align all of the pieces so that they fit when you go from one magnification to another. This is the revolving nose piece right here that has various lenses on them. So if you're interested, and I've had some students contact me, what do I need to look at for a microscope that I might want to buy online? And what I would do, it's better to have a slightly higher end microscope at the low end, if you know what I mean, than to have a low end microscope that's pushing the high end of it. You're going to be dealing with some cheap characteristics that it's kind of hard to avoid. So a nicer quality scope could cost around $200 to $300 at the low end, and you'll have a scope that has this revolving nose piece with various lenses on it. And usually a scanning lens, the smallest lens, it'll be 4x, and then the ocular eyepiece is up here. You see, it's a compound microscope. It has two lenses, and so the ocular piece is usually 10x across the board. Now, it doesn't always. I had a Zeiss microscope, and mine was 15x, the ocular piece, which gave it a little bit better performance. But the 10x, and then let's say we had a 4x scanning scope, which is the lowest, usually the lowest power objective on the revolving nose piece. So you multiply the two, and that gives you the final magnification. So if it's a 40x lens down here, or 4x, it would be 40 altogether. If it's a 40x lens here, then with the ocular piece it would be 400x. So you just multiply the 10x by whatever the power of the revolving nose piece is, and that gives you the final or total magnification. So scanning is usually, let's say, it depends on the scope that you have. It could be a 4x, it would be 40x, and it just allows you to find a position on a slide that looks interesting, and you can go after that. And then you have the body tube that holds the ocular piece. You could have one ocular or two. It's better to have a binocular type with two. It's easier on the eyes. A lot of times you can see an adjustment, and you can actually dial in your diopter for your glasses, and that corrects that, so you don't even have to wear glasses while you're looking through here, because the microscope, being an optic device, can compensate for your eye correction there, and boy, does that make life nicer. And then down we have the stage. Notice the clip. This is a clip that swings out and holds whatever slide that you have, and this is the back drop to it. And so you want the slide to move around on the stage. So you'll have stage manipulators, these on the side, that are adjustment knobs that move the stage. Now, you physically could try to put your hands up here and move the stage, but you can damage the gears. So it's best to use the manipulators on the side of the stage to move the stage. And really all it's doing is moving the hardware on the surface of the stage around so you can view what's on the slide. So this holds the slide, this metal piece here. This little flange right here moves out. You have a little handheld here, and you move this out, and this will swing out. You put your slide in, and it's sprung with a spring, and that will put a little bit of pressure on that slide and hold it in place. And then you move the manipulators, and you put the slide anywhere on the stage that you want. And on the stage we have these little numeric things that tell you where they are. So if, say, you ran out of time one day and you wanted to come back precisely where you were looking at, you can set it back to the magnification. We always record that, and then this will give you the XY coordinates on the stage, so you can go right back onto the slide where you looked. And so whenever we put a slide on the stage, we always put the label part against the backstop of the stage clip holder here, so you can see where the microscope slide would go. And it usually has a little white piece of tape or fritted, so that it's a different sandblast or whatever, and you put that edge against this side here, so you always put the slide on the stage the same way, and that way you don't have to worry about remembering those sorts of things. This adjusts. This is sort of, if you use cameras, this is your f-stop. You adjust the aperture, and that adjusts how much light actually gets in. The light source down here, and you can dampen this. There's various, depends on the scope. You can adjust the brightness by how much voltage goes to the light source here. Sometimes they have an adjustment that opens and closes an iris, so how much light gets through and that sort of thing, and it's there. So you usually have, between these two, you can adjust the amount of light that actually gets into the camera. Now the up and down, the focus, and that's save the best for the last. That's handled with the coarse adjust, which is always the larger knob, and then the fine adjust, which is the smaller knob. And adjusting these two, we can touch the coarse knob when we have the 4x lens. Anytime we go above that, we go to the coarse, the fine adjust knob, not the coarse, and adjust that. So it's the smaller knob. So once you do the official, as you get the slide on, looking at the scanning at 4x, you use the coarse adjust, and then anytime you move beyond that, you use the fine adjust, and you're good to go. And all that does, these knobs move the stage up and down. So these manipulators move it side to side, X and Y direction, and then the stage manipulators, as far as focus, it goes up and down. And I'll just tell you, as you go up in magnification and use the more powerful lenses, the less you want to move on the fine focus. There's terms for that, and we're going to go over it, but I just want to give you the effect. So you don't need to move very much when you get into the higher magnification. In fact, you want to move very slowly, because you can zip right through what they call the plane of focus, and not even aware to see it, and you'll be adjusting this until the cows come home in terms of trying to get it focused. So that's one of the biggest mistakes students make, and I will talk about that. So simple microscopes are ones that contain only one magnifying lens, and just you can get those 300x or so at the maybe a drugstore or something. So a magnifying glass would be called a simple one, and all it's doing is you have a lens and it's expanding the image, and so what they call a virtual image. So you're seeing the object, and it always inverts it. It's a characteristic of glass. Believe it or not, that the lens of your eye does the same thing. So the image for your eye, and you're probably already aware of that from anatomy, but the image that it forms on the back of the cornea of the eye is upside down. The brain knows that, and we see it as upright, but when you add an additional lens, it will flip that around, and so what we see will be upside down through a single lens system. And so I just wanted you to be aware. Okay, so the history of this, Lee and Hooke got the credit for it, even though there were others that were pretty strong in the day. And there's the stage on this little needle at the very end of it, and that is the lens right there. Believe it or not, it's just that little teeny piece of glass. And then you turn this up and down, and it's pathetic. But anyhow, hey, it was the best they had in 1600s, and that was great. His nemesis, this guy, Robert Hooke, was an amazing fellow. He was ruthless as a scientist. Oh, absolutely ruthless. I mean, what his is his, and what is yours is his. That's kind of how he viewed it. So if you did anything, he would claim it. And a lot of scientists don't like that, for obvious reasons. But Robert Hooke was really good with lenses, and he did things with microscopes that he learned from telescopes. A lot of Sir Isaac Newton's work, of course, in the days and looking at lenses and various things, and that technology kind of came first, and looking at things that are far away. And they said, wow, gee, I wonder if we can use this sort of technology to look at things close as smaller, and hence this is where we were. And Robert Hooke had some of that experience that he brought to the table. So that's a good thing, and we benefited from that. So Robert Hooke, though, he published a paper called Microscopy, and he drew things that you could see now with the use of the lens, and it was astonishing. People say, "Oh my gosh, you mean there's more detail and things smaller than we can see?" And yeah, so it's obvious to us, but it wasn't in those days. So here's the different types of scopes. This was the first one, and then they finally learned to put two lenses together. And so at some point in time, we had the two lenses, and it didn't really improve much for a long time. They just did fancy decorations on the body tube, and that's about it. And you notice the one, and then somebody said, well, why don't we have two, so we can look at it sort of like a binocular. And then that was sort of the next step up. And then they started to improve the optics with the lenses, and they went back to the single lens, because boy, those things would get expensive if you had two. And so that kind of had that period for a while, and then we came back to the two, like we typically have it at Wake Tech. We have the two, and then you might see a third in some of them, and that's where you put a camera, so you can actually visualize that. Mine's built into the camera, into the microscope, so I just plug a cable in here. But in the older days, they used to just put a regular type of camera and take pictures. You could do it that way. And so sometimes online you still see some of the older ones. It works if you have a camera to do that. So this is review a little bit. I just wanted to mention, though, the difference between the real image and the virtual image. It makes for good fodder for a test question. Magnification occurs in two phases. The real image is formed by the objective, the lens, the one that's on that revolving nose piece. And so the real image, the virtual image, is formed when the image is projected through the microscope body into the ocular lens. So the ocular lens is playing the magnification trick from the real image as a form of a virtual image. So it's the ocular lens that we use to manipulate, to create that virtual image. So the power of the objective, let's say we have 100x oil immersion types, which is about the high end, it's the highest magnification you'll get. The power of the ocular is 10x, so there's your calculation. So this is what I was blabbing on before. That's 1000x. If I added oil, that would add an additional. You just add 250x, that would be 1250x. It would be about the high end, topping off the scopes that we have in my scope here that I'll be showing for our labs. So now the only difference between this and my diagram, if it were me, this thing, I'm not sure, it's called a brain. I don't have that, but anyhow, you kind of work from there. So a light source, usually in a microscope, and you have a thyristor, something that makes this brighter, smaller, less intense, or whatever, and that goes up through the imaging part, which you could have an iris. I told you that's like an f-stop in the camera, and that condenses the light to make a nice beam that goes through your sample, your specimen, and then goes into the optics of the microscope. So the first, or the primary objective on the revolving nose piece, you're looking at the real image, and then as it goes up, there's usually a mirror or something that reflects. And this is something that Sir Isaac Newton worked out. It says, you know, optics can be battered by lenses. It's always good to use a mirror if you want to keep distortions out. So, and you can also make changes. You can go from 90-degree angles or whatever using mirrors, and there you go. And so this is all in the formation of what you perceive in the ocular lens. That is really what you're viewing is the virtual image. And so based on what the real image is providing through here, we use the ocular lens to tighten up on the virtual image. And then it's the quality of the lens system between the two that gives you the quality that you look for in like a Zeiss microscope, which is about the best in the world still to this day. So again, eyepiece up here, it's you can adjust this to correct for any changes that you may have for your glasses and things. There's the binocular body that holds it. Some of them spin around so you can have lab partners to look at it without moving the scope. You don't want to move a scope very much because vibrations and things can loosen and have an effect. The revolving or focusing nose piece that has the objectives on it, you can have 3 up to 4. I used a scope at a really nice facility at Emory University that actually had 5 objectives on it. It was amazing. I guess when you got lots of money you can do that. But so those are the objectives, and they printed right on them will be what they're able to do in terms of magnification. So you multiply that. Usually the eyepiece is 10, but they'll be written right on there. So 10 times whatever these are, and that'll be your final magnification, whatever you're looking at. And then the substage down here, we don't, once we adjust it, you kind of leave it alone. And so the iris diaphragm is sort of like the iris in our eye, adjusts how much light gets through, and the light source down here, on off, or sometimes it has a way of adjusting it. And that's pretty much the rheostat is how much power of the light. Usually it's on the side, or it can be on a separate knob. And then you have your coarse adjust, which will be the big knob, and the fine adjust will be the small. You always carry a microscope by holding the arm with one hand, and you put your other hand underneath the scope. If you drop it, obviously it's going to have a large effect on the quality of the scope from that period forward, as in no quality. So we don't want to do that. They're expensive. Now a good scope can run somewhere in the order of about $20,000-$30,000, maybe up to $100,000. I had a Carl Zeiss scope when I was at the vet school. That was a $100,000 scope. It wouldn't fit in my suitcase when I was leaving the school, unfortunately. I'm just joking. I would have loved to have kept that thing. So here's the lens, and all I wanted to demonstrate, I'm not interested in the physics, but the difference between the actual object and then the virtual image. So you may be looking at the image, but it's projecting it to the eyepiece, and then you see the virtual image much larger, and that's the key of a microscope. And it could take days to go over all the physics that's behind that, and I'm not interested in going through that here. I am interested that in resolution it's called resolving power. Now if you've ever taken an astronomy course, the resolving power is the ability to see a point of light or a star in the sky. If you can see the resolving power of your telescope, we'll see if it's 2 or 3 lights instead of just 1. So we're resolving that. It's the same sort of thing in a microscope. You want to look at, let's say, a cell. Is it one cell or is it multiple cells? And the resolving power will resolve it down to multiple cells if it's there. The capacity of an optical system to distinguish 2 adjacent objects or points from one another is the technical definition. So the resolving power of the human eye is about 2.2 millimeters. Mine's about 20 millimeters now. Age, I'm just joking. 0.2 millimeters. The resolving power of a light microscope using an oil immersion lens is about 2.2 micrometers. That's about 200 nanometers. So you can see that it's quite powerful. So the difference in low resolution versus high resolution, you can really start to see some detail, and which is important in science. We need to see those things. Using the refractive index is what they call that. We've all experienced it. You say, my God, what happened to the sun at night when it sets? It's this huge thing. Did it get bigger? No. The light rays actually bend out, and it gives you the perception that the sun is bigger because the light rays. It's a trick. It's a trick of atmosphere and moisture in the atmosphere. But we use the other way. We use oil, which bends light inward, and we'll get more light into the body tube. Hence, we get the better magnification because of the oil. So air tends to scatter it. The oil will bend it, and we'll get more into the body tube. So that's the key. Now, everyone's experienced this effect and having that refractive index. All you need to do You can convince yourself of this. Take a beaker that you have, take a pencil and just put it in the beaker and fill it up halfway with water, or so that way you'll have the refractive index of air versus water, and you'll see the pencil. And you follow the pencil up in the water, and then all of a sudden, boom, it's like half an inch over to one side. And I said, how is that even possible? And now you've seen an optical illusion based on the refractive index difference. So I challenge you to, if you haven't seen that already, you can see the refractive index and the tricks that can be pulled with that, and we just take advantage of oil bending light into where we want. So here at a simple thing with glass, and it appears that the straws over here, that's a refractive index trick. And so you can convince yourself. I'm not just drawing this and making hay and just, you know, to blab on about stuff. I want you to prove it. You know, put a pencil. It doesn't have to be a straw. You know. So importance of resolution is that you can see internal structures with better resolving types of scopes, and you can see resolve that, which is really important. And so you can determine if there's one or two cells there with a resolving power. Refractive index, if you want the technical terminology, is the degree of bending that light undergoes as it passes from one medium, now not a psychic medium being water or air or oil or whatever, and the higher the difference in the refractive indexes, the more the bending of light, which makes total sense, I hope. Too much light can reduce the contrast, and what it does is overwhelms the light. Now we all know about that. When the bright sun on, let's say, we get a reflection off of ocean water when we're at the beach, and you look at it and your eyes start to sizzle and burn and it hurts, and you can't really focus very well because there's just too much light. Well, what you can do is put on sunglasses, and boom, man, wow, everything, I can see everything. So that's kind of what you're doing, is reducing that amount of light, so contrast will improve. So iris diaphragm controls the amount of light coming into the condenser, so that we can cut down on the amount of light energy if we want to increase the contrast. And we won't have to adjust it much, but sometimes it gets moved around, and at least that's, you know what it does. The types of microscopy: a bright field, the most widely used type. It forms when a light is transmitted through the specimen, so we're using bright field. And the specimen, being denser, more opaque than its surrounding, absorbs some of the light, and the rest of the light is transmitted, so we see the contrasting as the two. This is for both live and stained and whatever. The bright field scope can be adapted as a dark field scope by adding a special disc called a stop to the condenser, and the stop blocks all the light from entering the objective lens except a peripheral light source, and so it sort of lights up around the edge, and then it's really cool. It kind of bleeds into what you're looking at, so it starts to glow and things get weird, and it sort of looks like moonlight. And anyhow, that's dark field, and the dark field has its purposes, and we can use those. Phase contrast, really cool. If you want almost a 3D experience when you're looking, phase contrast microscope will do that. Contains devices that transform the subtle changes in light as waves pass through the specimen. And what we do is it's the same thing as you go into a 3D movie and you wear some glasses that one eye has red and the other eye has green. What you're doing is you're changing the frequency by which the light goes through that filter. And the difference of the timing of the two creates this 3D effect. And so there's the notch. So that would be like the red side of your glasses, and this would be like the green side. And what happens ever so much difference as it goes through, the light will change its frequency just a little bit, its offset. And as a result of that offset, just like the different color glasses in 3D movie, you start to see things that appear in 3D. And that's really cool. It does look, it works, it's nice. Another trick that we can use is a fluorescence microscope. So its light source is UV. It's like walking into one of those stores that have black lights. And then all the lint in your shirt shows up, and as all the white clothes, it becomes really, really bright. Fluorescence uses a type of dye that is fluorescent stain, and if it binds to something, or if you take an antibody, which we'll talk about more later in chapters 12 and 13, we can put a tag on that antibody. And so if the antibody binds to whatever it is that it has high specificity for, we will now tag it, and it'll light up with color. You can see the image over here with the green, let's say, with green fluorescence. And you can use more than one dye. So you can have a fluorescent stain that's antibody raised for one thing and a different. It gets to be really fun. You can do all sorts of things, and you can do it in sort of a black field, so it just lights up using the fluorescence of the UV, and it's quite spectacular. Here's the scope, a UV scope we had at the vet school. It had post-processing effects with a computer and all that. This is about $200,000 scope. I used it a lot. It was a nice scope. I liked it. I love using fluorescence and tagging things. So you can see, yes, this is Bambizia or something like that. I can confirm it because it's stained green with fluorescence. And then, believe it or not, there's a white fluorescent stain, and I wanted to know where the chromosomes were and that sort of thing, and it'll pick it out. Or you can have different microorganisms and have a fluorescent tag on an antibody for a particular type of organism, and then have a different tag on another antibody that would bind to a different bacteria. So you see red and green, and I know that they're both there and what kind of the proportions. So it's nice. The scanning confocal microscope overcomes the problem of cells and structures by being too thick by using a laser beam of light, and this was kind of the first thing we used as a confocal before electron microscope, and it delivers a nice sharp image. The idea is it can get through your sample and you're able to capture a very nice image. I didn't use a confocal. It's either I used a light microscope or I used fluorescence, or then I just skipped right on to the scanning or electron microscope. So transmission and scanning are the two electron microscopes of prominence. And so TEM, which stands for the transmission electron microscope, is a very powerful microscope. It usually uses about 1,000,000 volts to generate the electrons that are zipping through that hit your sample. And what you're actually seeing, like this image down here, isn't the electrons hitting the sample, it's the electrons hitting the electrons of your sample, and they bounce off. And so where they bounce off is where we get to see the image, and it goes on a medium persistent phosphor screen, just like the old TVs we used to have. TV used to have a tube, and it had a sort of a cathode ray gun at the back, and it would spray the screen with electrons, and the phosphorus would light up if the gun hit it or it didn't, and it would create an image, and it would scan the whole thing before the phosphorus would wear out, and you would see TV. And yes, that's what they used to use. Today I know it's LED. I don't have a clue what planet is Dr. V from. I know I got all those comments, but that's what they used to use in TVs. I was the first kid on the block to have a color TV set because my dad used to work on TVs, and he got one, and it was pathetic. But anyhow, it did color, but not very well. But anyhow, boy, I was popular in that neighborhood. But anyhow, because electrons cannot easily penetrate thick preparations, the specimens have to be cut. So I used to earn a little extra money. I took an electron microscopy course, and then I'd earn just a little bit, and I got pretty good at it. And companies in the Research Triangle Park would pay me $500-600 a week to pull samples of various things, and that's how I helped try to pay for school. And what I would do, I'd spend most of the time preparing samples and slicing them with what they refer to as a diamond knife, and you make a cut so thin it's about I don't know, 1 to 2 angstroms, which were 100 nanometers. That's really, really thin. In fact, light bouncing off of that strip or the piece would shine gold. That's how you knew it was about that thickness. Anyhow, you float that onto your little grid and you put that in a TEM and a transmission scope, and you look at it. And you better be good, you better be fast, because it gets burned off in about, I don't know, 60 seconds, and then all that work is gone. So this picture was taken off of a screen during somebody's scanning event, and that's it. If you don't take the picture, it is gone. So the early scopes, which, believe it or not, I know, I'm not kidding you, I used a Microscope. It came over on the Mayflower. I mean, it's what we used to joke. You take a rubber mallet and you would hit part of the lens system. The lens system composed of these magnets that would adjust the electron flow. The electrons that would flow from the million volts being generated up here as it flies through. And so you would be looking at the lens here and then hitting various stages up here with a rubber hammer. And anyhow, these for the first one. Here's Ernst Ruska and his scope. It worked. I must admit it worked. As crude as it was, and State allowed students to utilize these scopes. And boy, you understood the basics of a transmission electron microscope. Let me tell you what, when you're dealing with these things, they are crude, I guess is the way to put it. So here's the more modern type. You have about 1 million volts. IBM built one that went up to 5 million volts, and it did no better than one with 3. So there is a maximum, just like light. Over a certain point, it doesn't offer much. And so about 1 million volts. And now what they use are coils that you energize with electricity, and they create a magnetic effect, and then that magnetic effect would tighten up the band of electrons as it goes through. Here's that grid I was talking about. This grid would be what you floated your sample on after you made that little cut, and it would float on this little copper grid, and you place that in the scope, and then all the viewing that goes in and the adjustment of the nice tight band of electrons would come and fly and hit your sample, and your sample would now eject the electrons onto the fluorescent screen, very much like that TV set that I told you about with medium fluorescent persistent phosphors. In other words, you hit it with an electron, it stays lit for a little while, and just long enough that you can kind of see the electrons being forced off your sample, and of course it maintains its relative position. And then you notice the cone of light. This is the key part. You notice the cone goes outward. That is how you experience the magnification right there. So you go from this smaller area into this much bigger area. So this is where you get the enlargement part. And I got some photos for you, but this is how an electron works, electron microscope. And this is what they call a transmission, because it's transmitting as a gun constantly through, and your samples being bombarded with these electrons, and then the sample is going to emit those electrons exactly from its position wherever it was, and then we magnify it by spreading that band out, and creates that image down here. And then you take you got a camera down here. In the old days it would just be old photographic paper, but now we use the same what we use with the cameras that we have in our phones, and it uses an LED receptive type, and it makes it a lot easier, cheaper too, to get those images. And so here's some transmission electron microscopes. This is a Joel. I used to use one like that. These are about $2 million. So here's this crappy photo of one that I took a snapshot over at the vet school. Sorry for the quality, but there you go. I just wanted to brag. That's the one that we used at the vet school. It was nice. It's a Philips. And we had another, this is one I grabbed, scarfed off the internet. I'm not really sure what the manufacturer is, but it's a nice one. You can see an older kind of computer setup, the old monitors and things, and then more modern, you have the setups. Here's a Joel that's this is about $1 million. It's got the source, and then you can use the setup. This is almost on the bridge of a scanning. It's a transmission. It uses a standard type, but you put your sample in through a door. These are the atmosphere that in which the electrons are flowing is a perfect vacuum. In other words, it's like space, deep space. There's nothing. I mean, we have iron getter pumps that pull out all the amounts of anything, so it has to be almost perfectly nothing in the atmosphere where these scopes work. I didn't mention that, but that's why it's so expensive. It has to pull it down. And this one is a little bit cheaper, and it was a Joel. I believe I hadn't used this one, but it's cheaper and it's on wheels, and you can always tell an expensive scope or not. You notice this one doesn't have wheels on it. It's sitting. It has to be in the basement of the building, that wherever it is on what they call ground rock, and so that's not moving in vibration. The slightest vibration could absolutely make your image blurred, and so that's kind of where we are. And so that's here is what you do to create the little slivers using what this is called a microtome. And this is a diamond knife that cuts your sample. So this would move up and down, and then the samples would be formed and come off onto water. And if you have this golden like sheen around your sample, then you take your grid and you pick that up with a grid and put that. This is right out of a TEM. This is where the electrons fly through, and you put your grid. This is made out of copper. Sometimes it's made out of gold, depends how deep your or how thick your wallet is. And then you put that on there, and the electrons would fly through it, and you would see your samples. You say, what kind of samples are we talking about, Dr. V? Well, I'm glad you asked that, because here we go. Here is a T6 phage, and that's slang, and you're going to know this semester what all that means. This is a T-even phage, bacteriophage. So these are viruses that attach to bacteria and inject its DNA into the bacteria and make more of these. And so that's what viruses do. So I'll show you a picture once I change this one. But this is just bacteriophages that we use a negative stain. It's usually, I forget the name of the stain. It's a dark stain, so like carbonized something rather. I forget what the name of it is. But anyhow, you use that and you get that. So here are the bacteriophages all over the surface of a cell. And it's a T-even phage, like T4. It affects, the T-evens affect E. coli. And they've attached so many, and then the bacteria cell becomes a manufacturing platform for the viruses once the DNA gets in there. And it makes more and more of them, so many in fact that it bursts the cell, and well, then it moves on to the next cell, and you get the point. Now this is all grayscale if you notice that, but we can use tricks now with the computers and colorize the grayscale. So you assign a color to a particular grayscale, and so it represents, and here's the rabies virus at 25,000x, and you can see it would be normally black and white or grayscale, and here you can see the genetic material, and they look like bullets. Rabies virus is creepy looking. It's just, rabies is just nasty all the way around. Of course it would look like that. It's just nasty. And you can quote me on that. So type of microscope, scanning. Now this is more affordable. This is a fun one. I played around with the scanning when the instructor wasn't around, but don't tell anybody. SEMs provide some of the most dramatic and realistic images. You don't have to prep them. You can just stick them under there. Unfortunately, whatever you stick in there, if it happens to be a live specimen, won't be too happy. It'll probably be killed by the time you finish with it, because the bombardment of electrons is a little rough. But SEM bombards the surface of the metal coated specimen, so you sputter a surface, some sort of metal on there, so it gives the nice electrons to be bounced off when you hit it with the flow of the scanning electrons. And so you get a really detailed look at this image. And so since it's been treated with that metal and you bombard it, every single position of that is accurately portrayed onto the imaging part. It works just like the transmission, but it's what they call scanned over a period of region, so you get a much bigger image, and much cleaner. And this was post-processed because still it's only grayscale, so they assigned a certain color. But I wanted to show you the detail that you can get with the scanning, and you get the amplification size, so with really good resolution. So this is kind of a picture of one that's a scanning, and you can see it's a much smaller tube. It's still got a vacuum pulled in here, but nowhere near to the degree that a transmission does. And these are manipulating your samples. You actually put your sample in over here, and this adjusts various positions of your sample. And this is the visualization. So you have some sort of electronic device that's probably on the back side over here, and it's the imaging, and this just takes that stream of electron image just like your computer monitor and displays it, and you can modify it and take pictures of the old days with a Polaroid. But what's happening is that you get the scanning rays, and then it strikes it, and then the electrons on the sample will bounce off, and that is what you see coming up on the screen. So it's really, really powerful. And so it's instant rate of the electrons flying off your sample, and it's going to do it wherever your sample's sitting. It'll be an accurate representation, but you're magnifying it by your coning out, spreading out the surface. This is kind of my feeble attempt to kind of show that. It's spanning it out, but that's where the magnification comes from, and so you get that. So let me show you an image. Here's a mosquito, and with it, if you can imagine I have a knob that's got numbers on it, and I just go click, click, click through it. So I just take this scanning EM, and I just go click, and I just zoomed in real time. This is about how fast it would be on a scanning electron microscope. And you click again, and I can really zoom in and zoom out. I mean, that is click, click, click. There you go. And that's and you can do this for a while, and eventually it starts to degrade, and bad things happen. So you can do that. So here's the head of a fly that somebody did and published it. I liked it because, boy, it was clean, very nice and clean. Here is a Ionodes scapularis tick, and notice the proboscis. This is what actually sticks into you, and he goes yum 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 with your blood. But these are little barbs. That's why it's hard to get these things out. And some say, well, if you take a match and you can tell it to close it. No, 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 no. What you want to do is let it stay. You take forceps and you go right across the upper part of the body, and you squeeze just as hard as you can and rip it off. Yeah, the proboscis stays, but you don't want all the bad stuff in its belly going into your skin or your body there. So if you have the misfortune of being bit, especially if you get a big red ring where it bit you, I would insist on a tetracycline treatment. Most doctors will do it as a prophylactic. Make sure you don't get Lyme disease. Here's a syphilis organism. It's a spirochete, Treponema pallidum. And the reason I showed this treponeme is the Lyme disease, Borrelia burgdorferi, is a spirochete as well. It's nasty. Some could say that it's a sexually transmitted disease, but it's not. It's tick vectored. But this one is sexually transmitted, syphilis, Treponema pallidum. And we're going to talk about this. What is really cool about microbiology is this outer part here spins, but the innards stay still. So it actually augers right into your skin. It spins, and wow, it's nasty. I could just get freaked out thinking about it. This is an organism that grows around thermal spouts somewhere in various places around the globe. And you can see all the detail. It's kind of spooky looking, but I couldn't find one that actually lives on our skin that looks very similar to this. So right now in your eyelids, you have millions of these little things actually has more teeth, and it's even scarier looking. But hey, I'll just leave it at that. All right, here's a crappy slide for you. It's an atomic force microscope. And this is the one I was talking about for the lab, about $20,000. It came down, and we could use atomic force at Wake Tech, but it'll be time. I have to write a grant to get that, but I think students would have a great time using this. How atomic force works is similar to transmission, but where you get your magnification and value is this little cantilever with a tip. That is where the nanometer, we look at things as a nanometer. And what we do, as this experiences the terrain and it's moving up and down, moving side to side, we hit it with a laser. So the laser hits down, and then essentially this moves around and it draws on a photoreceptor type plate. And then what you do is you get to see what this is actually drawing on or being pulled over. It does a really good job, but you got to drag it back and forth over and over. And we play games with math and things like that, and recreate the whole image as this is dragged apart. So you can see it's contacting it and it's moving up and down. It's basically just giving you an impression of what's going on with the image. And so the tip kind of is scraped over at the sample, and then the laser beam. I think you get the point. This is what you can see. And so, oh, what the heck is that? It's a little bit blurry. Well, what you're looking at is really at the atomic level. This is angstroms of things, and just what the chemists tell us, this is what we're looking at. So yeah, there you go. Yeah, we can actually see the rings and all the atomic structure. The chemists have been selling us for years. Yeah, we can kind of confirm that. So a little bit different from the standard light microscope. And so in the lab section, I'm going to be going over this scope, a little bit more detail, and how to prepare your specimens and all of that. And it's in your slide set. But instead of going over it twice, I'm going to stop here. It's been a fair amount of time, and I appreciate it's a little bit longer than I like, but I think you get the point of why I did that. And you'll get a lot more detail in the lab, and sort of a to kind of get you anticipating what the lab will be. And I go into a lot more samples and details of the light microscope and how to use it. So we'll be meeting at that point to try to get that lab started this week, so that you can start looking at and reading and doing things, and it'll be there for next week as part of its lab as well. And then as the kits come in, then we'll start rolling in. I'm hoping that next week we might start some of the kit work, and we'll see how it goes. Now, yeah, I'll just leave it at that, and hopefully things will get in. Right now we're at about 33% that have not gotten their kits yet, so I hope that changes over this week. So anyhow, that's it. You can look at whatever the exercises are. I have a few things, and a lot of this I'm just going to push into the lab part so you can do the exercises there. So, but this will be covered in the lecture part of the exam, and I'll have some practice questions, as I always do as a pretest. And talk to you soon.